This presentation is about proton pump inhibitors. Proton pump inhibitors are a treatment for GERD and peptic ulcers. GERD stands for gastroesophageal reflux disease. This disease occurs when food and gastric juices flow from the stomach into the esophagus past the lower esophageal sphincter. Peptic ulcers are sores in the stomach or duodenum caused by gastric acid. Risk factors for peptic ulcers include anti-inflammatory drugs, alcohol, caffeine, cigarettes, and radiation therapy. Also, high levels of stress, both physical and emotional, can lead to peptic ulcers. Each of these factors decrease the mucosal barrier in the stomach, in turn allowing for peptic ulcer formation. Elderly people, especially those with arthritis, have a heightened vulnerability for peptic ulcer formation. Also, those with a history of peptic ulcers or intestinal bleeding have a higher probability of recurrence. Due to a longer life expectancy, the prevalence of GERD-related symptoms is expected to increase in the future. Various medical conditions can increase the risk of peptic ulcers in GERD. These include Zollinger-Ellison syndrome, in which individuals have a higher gastric acid production. This disease also leads to the formation of tumors in the pancreas and duodenum. Hiatal hernia, which is present in 80% of people with GERD. Increasing body mass index has also been shown to be directly related to GERD severity. Hypercalcemia, which can increase gastrin production and can cause an increased stomach acidity. Scleroderma, which can harden the esophageal sphincter and can cause it to malfunction, allowing for gastric acid to enter the esophagus. And finally, the bacteria, Helicobacter pylori, which in studies has been deemed to be the cause of up to 80% of peptic ulcer formation. Some common symptoms of peptic ulcers and GERD include heartburn, regurgitation, and dysphagia, which is synonymous with a difficulty swallowing. Some less common features include chest pain, odynophagia, which is synonymous with painful swallowing, laryngitis, and dental hypersensitivity due to the erosion of enamel by gastric acid. If left untreated, peptic ulcers have various complications. These include internal bleeding, peritonitis, and scar tissue formation, which can partially inhibit the passage of food through the digestive tract. GERD, on the other hand, can lead to esophageal damage. Esophageal damage is the major complication of GERD. This damage includes reflux esophagitis, which is the deterioration of esophageal tissue and can lead to esophageal ulcers. Esophageal strictures, which is the narrowing of the esophagus. Barrett's esophagus, which is intestinal metaplasia and is precancerous. And finally, esophageal adenocarcinoma, which is esophageal cancer. Population data for reflux symptoms related to GERD are shown below. In a recent study of the weekly prevalence of GERD-related symptoms, Asia displayed the lowest percentage of symptomatic individuals, while North America displayed the highest percentage. Variations in statistics can be attributed to a few factors. The word heartburn does not directly translate into various languages, including Chinese, Swedish, Malay, and Korean. Racial distribution among population studies is not always consistent. These factors may in part explain the variations among GERD studies within specific populations.
Because excess production of stomach acid is the root cause of GERD and gastric ulcers, reducing gastric acid production is the primary goal of drugs designed to treat GERD. Gastric acid is produced by the cells that make up the lining of the stomach, which are called gastric parietal cells. The gastric parietal cells have a transmembrane ion pump known as the gastric hydrogen potassium ATPase. This protein pumps potassium ions into the cell and protons out of the cell into the stomach. Because these ions are being pumped against their concentration gradients, this process requires energy, and the pump hydrolyzes ATP to fuel the process. The action of this pump is mediated by acetylcholine, histamine, and gastrin signals that come from the brain and other nearby cells in response to the state of the stomach. More stomach acid is secreted when you eat things that need to be broken down. There are several approaches to reducing the amount of stomach acid a person secretes. The simplest solution is to take antacids, which are basic compounds that neutralize the acid in the stomach. Tums are an example of an antacid. Antacids work quickly, but have to be taken every time a person experiences symptoms and do not treat the root cause of excess acid secretion. The first class of drugs designed to treat the cause of acid secretion were the H2 receptor antagonists. The H in H2 stands for histamine, and recall that stomach acid secretion is mediated in part by histamine. The H2 receptor antagonists block histamine signaling to indirectly inhibit gastric acid secretion. In recent years, proton-pump inhibitors, PPIs, have largely replaced H2 receptor antagonists as the treatment of choice for GERD. PPIs directly inhibit the gastric proton pump, stopping acid secretion. As we will see, they have proven to be more effective than H2 receptor antagonists. So let's talk a little bit more about the target of proton pump inhibitors. Again, an ion pump called gastric hydrogen potassium ATPase is responsible for secreting hydrogen ions into the stomach and lowering its pH. The diagram on this slide illustrates the process. The gastric proton pump uses the energy gained from hydrolyzing ATP to ADP to transport sodium ions from the stomach into the parietal stomach lining cells and moves protons in the other direction from the parietal cells into the stomach. The net effect of the action of this proton pump is the accumulation of hydrogen ions in the stomach, which lowers the pH. This allows for the digestion of food, but in excess can cause the symptoms of GERD and peptic ulcers that we described. Proton pump inhibitors inhibit the action of this protein to reduce the acidity of the stomach. So what does a PPI look like? This slide shows the general structure, or pharmacophore, of a proton pump inhibitor. PPIs consist of three major elements: a benzimidazole ring and a pyridine ring linked together by a sulfoxide. The benzimidazole and pyridine rings are functionalized with R groups, and the identity of these R groups distinguishes the different PPIs. Now let's discuss how the PPIs act on the gastric proton pump. PPIs are administered as prodrugs and must first become activated in the body before they can inhibit the proton pump. There are two steps involved in the activation of a PPI. First, the benzimidazole ring is protonated, and second, the sulfoxide rearranges into a sulfenic acid. After the drug is activated, it can form a covalent disulfide bond with the proton pump, inhibiting it. Let's look at these steps in detail. In the first step, the benzimidazole becomes protonated. This is illustrated here, in red. There are other protonated forms where the pyridine is protonated instead that exist in equilibrium with the protonated benzimidazole, but we have omitted those because they are not directly active in the mechanism of inhibition. Next, in a two-step rearrangement, the sulfoxide becomes a sulfenic acid, which is a sulfur bonded to an alcohol. To begin the rearrangement, the lone pair on the nitrogen of the pyridine ring attacks the double bond on the protonated, positively charged nitrogen of the benzimidazole ring. The double bond collapses onto the nitrogen, satisfying its positive charge, and a tetracyclic intermediate is formed. Then, the hydrogen attached to the benzimidazole leaves, and the electrons collapse into one of the nitrogen's bonds, reforming a double bond that was removed in the first step. When the double bond reforms, it breaks one of the adjacent bonds, pushing it up onto the sulfur atom. This, in turn, pushes electrons from the sulfur-oxygen double bond up onto the oxygen, which becomes protonated to satisfy its charge. The end result is a charged compound with a sulfenic acid group, and this is the active form of a PPI. The activated PPI, in sulfenic acid form, is a very thiophilic species, meaning that it wants to bond to another sulfur atom. The gastric proton pump, like almost all proteins, contains sulfur in its cysteine amino acid residues. In a dehydration reaction, the sulfur from the sulfenic acid forms a disulfide bridge with the sulfur in a cysteine residue of the gastric proton pump, forming a covalent bond between the PPI and the gastric proton pump. The formation of this bond inhibits the proton pump. So why are PPIs effective against only the gastric proton pump? Why don't they bind to the cysteine residues present in almost every other protein? The answer lies in the acidity of PPIs. They are administered orally and absorbed in the small intestine like most drugs. Appropriately, they are delivered in a capsule with an acid-resistant coating, so they survive the intense stomach acid they are intended to treat and make it to the small intestine for absorption. Due to their acidity, PPIs concentrate in acidic intracellular spaces. In particular, the area near the gastric proton pump is especially acidic. Since the gastric proton pump moves hydrogen ions out of the parietal cell, there is a high concentration of hydrogen ions localized to the area immediately adjacent to the pump, where they are being transported across the membrane into the stomach. This makes the secretory area around the gastric proton pump a particularly acidic space, and PPIs accumulate here. In fact, PPIs are found in the secretory area of the proton pump at a concentration over 1,000 times higher than in the blood. In this acidic environment, the PPI can also undergo the protonation and rearrangement required to activate the drug. So we now have a very high concentration of drug, higher than anywhere else in the body, directly localized next to its target, in conditions that are necessary for its activation. This is how PPIs react only with the gastric proton pump, and not with other cysteine-containing proteins. The activated PPI binds to one of several cysteine residues on the gastric proton pump, forming an irreversible covalent bond. Because this bond is covalent, PPIs can be active for a relatively long time, over 24 hours, despite their short half-life of only 60 to 90 minutes in the blood. This is a picture of the crystal structure of the gastric proton pump of a pig. The pig gastric proton pump has a high degree of homology to the human version. The long section near the bottom of the molecule, made up of many aligned alpha helices, is the transmembrane portion of the protein. It's near these transmembrane domains that PPIs bind to cysteine residues. Formation of the disulfide bond to the cysteine residues inhibits the proton pump. The pump has to undergo several dramatic shifts in conformation during the process of pumping ions. The attachment of PPIs to the cysteines prevents the pump from undergoing these conformational changes. This is because the PPI, which is now attached to the pump, cannot move freely through the membrane as the pump shifts because it is not lipophilic enough to interact with the membrane due to its positive charge. Thus, attachments of the PPI blocks the conformational shifts of the pump, inhibiting its action and preventing the pumping of hydrogen ions into the stomach.
So now we will go over the discovery of the first PPI drug, Omeprazole. H2 receptor antagonists may be considered the first generation of drugs used to inhibit gastric acid secretion. In fact, the first blockbuster drug was actually cimetidine. The problem with H2 receptor antagonists is simply that relatively high or frequent dosages are required because of short duration of action. H2 blockers have a quick onset of action in less than an hour and have peak activity with 3 hours, but after 12 hours they are completely used up. But a different class of anti-secretory drugs began to develop in the 1980s that would improve greatly on this deficiency in activity, PPIs. In the late 1960s, a research project began in a division of the drug company Astra in Sweden. Now it's AstraZeneca. The first goal was to develop an orally bioavailable anti-secretory drug to fight peptic ulcers. So this was a target-based drug discovery initially. Researchers tried to use local anesthetics to target the antrum of dogs to block the release of gastrin. However, the acidic environment of the stomach would protonate and deactivate the drugs. The next goal was to make lidocaine, an anesthetic that Astra acquired rights to several decades earlier, non-basic in order to prevent protonation. Several compounds were synthesized to this effect, but were generally toxic when tested in vivo in rats and dogs. But one type of drug, the carbamate, was hopeful because of its effectiveness in rats. So, research began to wane and the group was losing funding. They ran a literature search and found CMN131, a potent anti-secretory thioamide from another drug research group. It was toxic and research had been called off. Astra wondered at the causes of toxicity and hypothesized that the double-bonded sulfur was the problem. So they decided to take CMN131 and incorporate its thioamide group into or between heterocyclic rings to hopefully reduce its toxicity. With this new goal in mind, a phenotypic drug discovery process began. Many benzimidazoles were synthesized and screened in biochemical assays. The first hit was H12426, and it would have been the first lead if not for a Hungarian patent that covered the compound and its patent for tuberculosis drugs. Okay, so the first lead was a failure because of patent issues. But Astra did not give up and collaborated with Abbott Laboratories to continue research by studying the metabolites of H12426. Two main metabolites were found to form rapidly. H8369, the compound sulfoxide, was adopted as the first lead and called Omeprazole. There were no intellectual property right conflicts over this one. Unfortunately, again, Omeprazole was toxic in vivo. It seemed to inhibit iodine uptake and caused atrophy of the thymus gland. Another literature search indicated some substituted benzimidazoles don't affect iodine uptake. One particular compound that was discovered, H14994, displayed high activity without inhibiting iodine uptake. It would later be named picoprazole. Picoprazole became the first successful lead, lacking in toxicity and high in potency. In vitro models were designed around that time for better optimization. As picoprazole was being optimized, the hydrogen potassium ATPase pump was discovered. From this point on, PPI drug discovery became target-based. The final steps of optimization included making picoprazole more basic, and the final result was the first PPI drug, Omeprazole. In the change from picoprazole to Omeprazole, the pKa increased by a factor of 1, going from about 3 to 4 in Omeprazole. This is finally some great news. An investigational new drug application was sent out in 1980, and soon it became a clinical candidate. Clinical trials began 2 years later. It was found to be non-toxic, though with some adverse effects admittedly, and it was superior to the leading H2 antagonist of the time, ranitidine. In 1988, Omeprazole was released in Europe as Losec, and in 1990 it was released as Prilosec in the United States of America. Now, let's go over some pharmacokinetics of Omeprazole and some other PPIs that have come about since then. Omeprazole is orally administered in enteric coated granules that allow the drug to make it to the hepatic portal system. The time of maximum concentration is a few hours after administration, and the volume of distribution in the average person, around 70 kilograms, is from 9.1 to 24.5 liters. So the drug tends to leave the plasma, in this case to accumulate at the acidic environment around the parietal stomach cells. While the drug itself has a considerably short half-life in the plasma, the fact that it irreversibly binds to the proton pump makes its effective half-life of activity much longer, around 24 hours. For most PPIs, absorption, distribution, metabolism, and excretion are generally the same. In absorption, the oral bioavailability of omeprazole is 40 to 50%, and it's higher than that for some other PPIs. This is fairly low due to first-pass metabolism by the liver. For distribution, PPIs are mainly distributed in the plasma, not in blood cells, and all of them demonstrate greater than 90% binding to plasma proteins. For metabolism, they are rapidly metabolized by the liver from, principally, phase 1 oxidation by cytochrome P450 2C19 and cytochrome P450 3A4. One issue with the pharmacogenetics of this drug is that some subpopulations lack cytochrome P450 2C19 and are therefore slow metabolizers of some PPIs like omeprazole. Finally, PPIs are mostly excreted in the urine from the kidneys, and small amounts are also found in the feces by the work of the gallbladder. It turns out that omeprazole, due to low bioavailability, also required higher than recommended doses, so in 1987, Astra synthesized and screened hundreds of compounds to improve this bioavailability problem. The only hit to become a lead among them was the S-optical isomer of omeprazole, called esomeprazole. Esomeprazole was launched more recently by AstraZeneca. It has a higher oral bioavailability because the first-pass metabolism is stereoselective. We can see from other PPIs that have been developed that doses, area under curves, volume of distributions, half-lives, and clearances are very similar for each drug in the class. Tanatoprazole, a recent drug development, could be useful for nighttime treatment because of its exceptionally high half-life of 8.7 hours. We can also see that esomeprazole does have higher oral bioavailability than omeprazole. Dexlansoprazole and rabeprazole are still relatively low in oral bioavailability. Presented here are the structures of these other PPIs. All of them follow the previously shown pharmacophore except for tanatoprazole. We have lansoprazole, rabeprazole, pantoprazole, esomeprazole, dexlansoprazole, and tanatoprazole. Tanatoprazole is an imidazopyridine derivative, rather than a benzimidazole derivative. Because a nitrogen atom has replaced a carbon-hydrogen bond in the main benzimidazole group, tanatoprazole could be considered a classical isoster with other PPIs. Finally, all current PPIs have similar adverse effects, like possible decrease in absorption of magnesium or calcium, headaches, and nausea. Esomeprazole and dexlansoprazole have the greatest activity among the PPIs on the market today. Despite their possible adverse effects, PPIs have been shown clinically and on the market to effectively and safely reduce gastric acid secretion